All right, welcome everyone. I'm Kunal Ghosh, founder and CEO of Inscopix. Welcome to the second Inscopix Insights webinar. I see that some of you joined us 2 weeks ago or are joining us again, and welcome back. And for those of you who are joining us for the first time, you're in for a treat with Dr. Tom Cash today being our second webinar speaker. So welcome to all of you. Certainly hope that all of you and your loved ones are doing well and staying safe as we continue to tackle this pandemic. As I'd mentioned in my earlier remarks during David's talk, the Insights webinar series is an effort by Inscopix to do our best to keep the neuroscience community hopeful, vibrant, and socially connected as we continue to practice physical distancing. With a spectacular lineup of world-class neuroscientists, the series was kicked off by Dr. David Anderson of Caltech a couple of weeks back. And today we will have Dr. Tom Cash from the University of North Carolina, Chapel Hill. It's my absolute honor and privilege to welcome Tom. Tom is the John Andrews Distinguished Professor in the Department of Pharmacology at UNC Chapel Hill. The broad scientific goal of Tom's research is to, at the end of the day, understand how modulation of discrete neuronal circuits can shape behavior and to deconstruct the molecular mechanisms that underlie that modulation. Amongst Tom's many honors and awards, Tom is the recipient of the White House PCAS, Presidential Early Career Award for Scientists and Engineers Award, and is also known for seminal contributions with respect to understanding stress and how stress and drug abuse can alter neuronal function in brain regions that regulate emotional behavior. So today, Tom will discuss his efforts and those of his groups, of course, in attempting to understand how neuromodulators such as peptides and catecholamines can alter cellular function and change behavior. His talk will focus specifically on the BNST, the bed nucleus of the stria terminalis, a brain region that is well known to regulate a host of emotional behaviors. So with that, welcome, Tom. The virtual floor is yours.
Great. Well, thank you, Kunal and the Inscopix team for having me give this talk. It's great to have the opportunity to share some of the data from the lab. And so Kunal hit upon really what we're interested in the lab is trying to understand how modulation can impact circuits. And really kind of the issue that we focused on for many years is understanding substance abuse and kind of comorbid disorders. In particular, one thing that really has drawn my attention is the trying to understand the interaction between aversive states such as anxiety, pain, dysphoria with reward or drug consumption. And this is interesting to me because this is a bidirectional interaction. Aversive states can influence drug consumption, but drug consumption can also influence these states. And so this is actually a perfect use of animal models, is dissociating this complex bidirectional interaction. Today, what I'm going to talk about is a brain region called the bed nucleus of the stria terminalis. And while this is not the only region involved in this interaction between stress and negative aversive states and reward, it is an important one. And so the BNST, as I'll refer to it, is a part of the extended amygdala. It receives a dense input from another part of the extended amygdala, the central nucleus of the amygdala, or CEA. It also receives glutamatergic inputs from the basolateral amygdala, and as well as glutamatergic inputs from cortical, hippocampal, and thalamic regions. Thalamic region is not shown on this graph. And really, to me, where the rubber hits road for the BNST is that there's a series of these bidirectional interactions with hypothalamic, midbrain, and brainstem regions. And it's through these bidirectional interactions that the BNST is well situated to control brain-wide networks and brain-wide states. And so this is something, this topic has been explored deeply for the past 50 years at least, through behavioral pharmacology as well as other assessments. And just from kind of highlighting what I view as important for how we're approaching this, the structure plays an essential role in stress-induced reinstatement, opioid withdrawal-induced aversion, sustained fear or anxiety. This is from Mike Davis's group, as well as an emerging, really elegant data set from the Manami lab looking at pain-related behaviors. More recently, we and others have found that circuit manipulations of this region can impact anxiety, reward seeking, stress coping response and alcohol consumption. This is not an exhaustive list. There's been a number of really outstanding and exciting studies coming out every week. This is just kind of a few that we want to highlight that I think are important here. And so this is not just a rodent brain region. This is a region in humans that is important as well. Here is a fMRI study from the Blackford group at Vanderbilt, just showing that with an unpredictable threat, you see that the BNST is selectively engaged in people. And so this highlights that the BNST is involved in unpredictable or ambiguous threats that are correlated with heightened anxiety and arousal in people as well. So I think this has a lot of, this is an important region that's small but mighty. And the way that I've approached this work has been trying to understand how modulation works. And just a step back as we think about modulation, we think it's important because this can shape neuronal function and behavior on multiple scales. And so if we think about, here's a neuron, just kind of your run-of-the-mill, we'll call this an output neuron. These neurons receive a GABAergic or inhibitory input, and then excitatory or glutamate input. And so there's a body of data that suggests the balance and timing of excitation and inhibition can have a large impact on information flow through circuits. But layering atop that, we have modulators. And so these are compounds like neuropeptides, such as corticotropin releasing factor, CRF, or biogenic amines like serotonin or dopamine. And how these work are through the actions signaling at GPCRs, or G protein coupled receptors, which can be located at multiple points within this circuit. They can be located on cell bodies, they can be located on terminals. I didn't show it in this diagram. We can also have, these are loaded up on astrocytes as well. So these are here to tune the function of these networks. They can excite excitatory inputs, inhibitory inputs, and kind of overall change the gain of the system in interesting ways. And the reason we're interested in this is we think this is important for determining, can play roles in pathological function associated with brain disorders, as well as they're implicated, drugs act on GPCR. So this is a way to normalize function within circuits. And so the BNST is an interesting region from the standpoint of modulators, because this is really a modulatory soup. There's a host of neuropeptides and biogenic amines in this region. So this is just some of the compounds that we've studied over the years, looking at them. And today, what I'm going to hone in are two particular compounds that we think may interact in an interesting way. Dopamine, shown here with DA, and CRF, corticotropin releasing factor, CRF. And so what I'm going to talk to you today, so this is all actually, or 95% unpublished work. This is the work of Weilin Yu, who is a graduate student in the Cash Lab, who is actually going to hopefully graduate this September. I say hopefully because we're trying to determine timing, as we're not, we're all doing this thing. But there are two parts to what he's looked at. Part one is going to focus on a population of dopamine neurons and their connectivity with the BNST. And part two is going to focus on CRF signaling and CRF neurons within the BNST and how they relate to pain-related behaviors. And so what we know about dopamine signaling, so focus on dopamine first. We know that dopamine signaling in the BNST is associated with opioid, alcohol seeking, and pain. And here's just a few citations highlighting this. So this obviously isn't as well known and well studied as, for example, dopamine signaling in the nucleus accumbens, but that doesn't mean it's not important. So these studies have kind of implicated it in a range of processes, range of behavioral processes that we're interested in. And what is interesting to me, and this is work that I actually started when I was a postdoc in Danny Winder's lab, was that the dopamine inputs to the BNST are varied, we'll say. So there is a dopamine input from the ventral tegmental area. This is kind of the classic dopamine region that people think of. But there's also a dopamine input from the ventrolateral PAG / the dorsal raphe, depending on how you want to call it. I'll probably refer to it mostly as the PAG. And so these inputs from the PAG really early on piqued the interest piqued my interest and the interest of the lab. And so we know that these receive inputs, and if we look at the PAG, and this is using a TH reporter mouse, there are TH positive neurons in this structure, the PAG. Moreover, if we use a genetic tracing method, we can see that they provide a dense input to the dorsolateral BNS2 well. So there's this, I would say, moderately dense input from these dopamine neurons in the PAG. But what do we know about them? And so classically, the ventrolateral PAG is a critical site of action for the analgesic actions of opiates. And this is, an immense body of work from many labs, including Susie Ingram, Howard Fields, too many to mention. But furthermore, these neurons, some early work from Lou and Saper found that these are implicated in sleep-wake behaviors. These neurons also appear to be involved in reward and pain relief properties of opiates, studies from the early 2000s. And then what we've done since we've been in the lab, and cash lab at UNC, is we've looked and found that these neurons are activated by both alcohol and opiates. And so an interesting thing about these neurons, so initially we wanted to look and see what these neurons could do downstream. We used a channelrhodopsin-assisted circuit mapping approach, injected a TH-Cre mouse with a Cre-dependent channelrhodopsin, and then waited 2 months and recorded from neurons within the BNST, and specifically we're focusing on the dorsolateral BNST for the aficionados. And we found that there was this short latency release of glutamate. So these appear to have glutamate as a co-transmitter, but also using fast-scan cyclic voltammetry, and this is a collaboration with the McElliott lab, we found that there was evidence for dopamine release. I would say that others have found this, specifically the TILAB published a paper a few years back that showed similar properties of neurons. And we've also found that VGLU2 is co-expressed in these TH neurons. And so these neurons have this interesting co-release property that is not uncommon among midbrain dopamine neurons or midbrain neurons, but what about functional properties? And so the very first experiment we did was just look at what happens when we activate these neurons using a chemogenetic approach. And this is using a DREAD approach pioneered by my collaborator and colleague, Brian Roth, here at UNC, which allows us to chemically or genetically express a GQ-coupled DREAD. And so this is a designer receptor exclusively activated by a designer drug that lies in rest until you give this designer drug. In this case, we use CNO, and you can see that CNO excites these neurons. And when we give CNO to awake behaving animals, we focused on pain-related measures. And very first measurement was we just looked at the latency to withdraw a paw on a hot plate. So this is a test of thermal nociception. And when we activate these receptors in TH mice, you can see, in TH neurons rather, you can see that there's an increase in the latency to paw withdrawal. So this is an anti-nociceptive response, but we did not see any effects on tail flick or locomotor activity. And so this was our initial study. And then when Wayland joined the lab, so this is kind of the state of the knowledge, and I would say that a recent study that was published in eNeuro found very similar results using a DAT-Cre mouse line. And so this was exciting to see that another group had generated, I would say, a complementary and synergistic data set, reiterating what we had found. One of the things we were interested in, though, when Wayland joined the lab, he wanted to follow up on these studies. And we decided, because it had been a few years since we had done these specific behaviors, that we should revisit this and do so in a rigorous fashion. And so the question that Wayland asked was, does this ability of this GQ DREAD, the ability to alter pain-related behaviors, does it depend on sex? So we did it in male and female mice, or does it depend on the pain state? And so we used a model of persistent inflammatory pain. And so the first set of experiments, the approach we used was nearly identical. We injected a Cre-dependent HM3D cure control into TH-Cre animals, waited several weeks. We conducted pain sensitivity testing. And in this case, what we did, instead of using a hot plate, we used a Hargreaves assay, which is another test of thermal sensitivity, thermal nociception, as well as a von Frey, which is a test for mechanical sensitivity. So we did these kind of, I would call them baseline pain sensitivity tests or measures of behavior, and then treated a paw with CFA, or conditioned Freud's adjuvant, which induces an inflammatory state, which makes the mice have a greater sensitivity to these assays, and then conducted the same sort of testing where we activate these neurons and then sacked the mice and then conducted a histological validation. And so when we did this, or when Wayland did this, what he found was that, so we're looking now in male mice, and so I'm just showing the saline and CNO comparison. Focusing on the CNO, you can see that when we give CNO to these GQ-DREAD mice in red, there is an increase in Paul withdrawal latency. So this is the Hargreaves approach. So an increase in the amount of time on this indicates an anti-nociceptive effect. So they're able to keep their paw in this heat exposure longer. So this is consistent with what we found previously. However, when we looked at the female mice, we saw a pretty much a non-effect, we'll say, with this assay. And so this was interesting. And perhaps when you look at the literature for the pain field, not entirely surprising to see a sex difference within the PAG. And there's really beautiful work by Anne Murphy and others highlighting how the PAG is this critical nexus of sex differences with regards to opiate-related pain behaviors. So this was somewhat surprising, but maybe not unexpected. So this was with thermal pain or thermal measures. And so we then repeated this for the mechanical assay. This is a von Frey. And again, we see that activation of these neurons can increase the mechanosensitivity threshold. So again, this was anti-nociceptive in male mice. And when we looked at female mice, we found no significant effect here. And so I'll come back to this, so hold that in your head for 1 second. But next, what we wanted to ask was, okay, we see this seemingly sex-dependent effect where there's an anti-nociceptive effect of activation in male but not female mice. What about following treatment with this, inducing a persistent inflammatory pain state? And so what we did, we conducted the same study, and in males, we saw again that this was able to induce an anti-nociceptive activation of these neurons to induce an anti-nociceptive response. Again, in female mice, we saw that there was no effect, no difference in behavior upon activation of these neurons. In male mice, looking at the mechanical sensitivity, again, we see that we're able to, activation of these neurons is anti-nociceptive, whereas in the female mice, there is, again, no impact on this behavior. And so this was, again, perhaps this makes sense, but something when we did this initial study, after we were looking at it for a bit, this initial DREAD study just in baseline mice, we wanted to ask the question, we noticed that there was a fair amount of variance in the response to CNO in female mice. And so we wanted to ask the question, does estrus cycle impact GQ-DREAD-driven antinociception? And this is important for a host of reasons, particularly, estrus cycle can impact many of the circuits we're focusing on and also dopamine signaling. So it made sense to do this. So we injected a series of new mice and then conducted the same assays and then did this intermittently over several weeks. And then after each assay, what Whalen would do is assess estrus. And so when we did this, again, we saw notably no effect in the Hargreaves. And there's obviously no estrus effect for this no effect in Hargreaves. When we looked at the von Frey, what we saw was that there was a significant effect when we ran a two-way ANOVA here of the HM3DQ of the virus. And so the p-value here is 0.0503. And you can see that there is this, with this repeated testing paradigm, we saw an anti-nociceptive effect in the von Frey assay. However, it really didn't have anything to do with estrus. And so for us, this was interesting because what it did was it suggested that, we still are seeing this variance in females, but it perhaps has nothing to do with estrus or, not the way we're looking at it. So this is kind of an ongoing question for us. And so this, again, so this is something that we're still thinking about and trying to interpret these studies or, to the next step for these studies. But the next question we had, what role is dopamine playing in these behavioral changes? And the reason this was important to us was that, or an important question to ask, was that these neurons can co-release glutamate as well. So how do we know that it's not glutamate release from these neurons that's driving these effects versus dopamine release? And luckily, we had a way to address this, I would say, relatively easily. And this is a collaboration with this Weifel lab at University of Washington. And he recently developed a series of single AAV-driven CRISPR constructs that allow knockdown of discrete genes. And luckily for us, he had one for the for tyrosine hydroxylase, the rate limiting step for dopamine synthesis. And so we got in touch with Larry, got the knockdown vector or the TH CRISPR vector and the control vector, and then injected mice, and also injected the mice with a Cre-dependent HM3DQ. And the idea here was, we know that chemogenetic activation of these neurons can have these effects on behavior. And so are they dependent upon TH signaling, or dopamine signaling is how we'll interpret it. And so you can see these vectors work well. We get about a 50% or so knockdown in males and females. And then the experiments we did, we just, in this particular set of studies, we just looked at thermal and mechanical nociception. We didn't treat the mice with CFA. And so what we found was that, again, so now we're looking at the von Frey assay. And I would say, focusing on the dark green bars, you can see that CNO is able to increase the mechanical sensitivity threshold. So this is an anti-nociceptive response with HM3DQ and the control vector. However, when we use the TH CRISPR vector, this response is blocked. And so we no longer see this response for the von Frey assay. And then again, in females, you can see that we see no response for the HM3DQ plus control, nor do we see a response for the CRISPR. And we have some ideas why we might not be seeing a response in this particular set of experiments that I'm happy to talk about later. It's kind of a little bit of an aside. And we also looked at the Hargreaves approach, assessing thermal response to a thermal stimuli. And again, we see the same sort of response, that the HM3DQ with control vector is anti-nociceptive, whereas when we combine this with the TH CRISPR vector, this blocks this effect. And again, we see no effect in females. And so taken together, this tells us that the chemogenetic activation of these neurons are impacting behavior, likely in part through dopamine signaling. And so then the question we had, we kind of started this out thinking about the BNST. This is where this project started about 12 or so years ago when I was a postdoc, but we really haven't addressed that interaction yet. And so the question Wayland asked was, what if we photostimulate these outputs to BNST? What impact does that have on behavior? And so what Wayland did was inject Cre-dependent chanorodopsin into the PAG, put bilateral optical fibers over the BNST, and then just photostimulate. And we picked 20 hertz as our stimulation frequency. And so what we found was that, again, we're looking at the Hargreaves assay. Now this is a measure of thermal response. We can see that stimulation of these inputs is anti-nociceptive in males, however not in female mice. And then when we looked at mechanical thresholds, we see again that this is anti-nociceptive in male mice, but not in female mice. And so this is broadly consistent with what we saw with the GQ signaling, the chemogenetic signaling. I will kind of add as a post hoc, some of the ends for the female mice are slightly lower, in particular the controls. We have 4 in one case. So we're likely, I think what we have to do is increase the sample size to at least balance out these groups. And so this is something that we'll do eventually when we can go back to work. And so this, again, was interesting, and it kind of led us to ask the question, what is going on when we stimulate this? Can we measure the responses in the BNST? And so are there sex differences in this optically evoked, this synaptic transmission, this pathway? And so we went back to this chanorodopsin-assisted circuit mapping approach, injected AAV into the PAG, these TH Cre mice, waited a couple months, and then recorded in the BNST. And so notably, what we saw, again, there is this short latency optically evoked EPSC that we know is due to direct release of glutamate. However, what I didn't mention before, but we also see, is this longer latency polysynaptic optically evoked iPSC. And so we know this is polysynaptic because when we put on tetrodotoxin and 4-AP, this response goes away. However, the glutamate response persists, and it's blocked via GABA A receptor antagonist. So we can use this approach to measure both optically evoked monosynaptic responses of glutamate and this polysynaptic GABAergic response to give us some sort of understanding or idea about what's happening within the microcircuits within the BNST. So when we did this, we measured a number of different properties, just highlighting a few of them, we looked at the amplitude of this glutamate event and saw no significant difference between sexes. We looked at the amplitude of the iPSC, again, saw no significant difference between the groups. Looked at the paired pulse ratio. This is, can provide some information about the probability of release. We saw no effect for the glutamatergic events. When we looked at the paired pulse ratio for the males and females for the GABAergic responses, we saw that there was a significant reduction in the paired pulse ratio in females compared to males. And so we're still thinking about how to interpret this. And I think what it might highlight is that the GABAergic microcircuits that are engaged by this stimulation might have fundamental biophysical properties that are different between males and females. And this is something we have to suss out, I think, a little bit more using other approaches. And so another thing we can do with this data set is just look at the distribution of responses. And so what we did was just look at cells that responded versus did not respond. And so when we look at glutamate transmission, we see in males, we get about half the cells respond with an optically evoked EPSC. In females, we see a similar value, about half the cells respond. So there's no significant difference there. But we did see something interesting when we looked at this polysynaptic iPSC, in that in males, we saw about 40% of the neurons showed this polysynaptic inhibitory response, whereas in the female mice, we only saw 15%. So it kind of layers onto this idea that there might be some differences in these local, the ability of this input to engage local circuits. And that might be via glutamate signaling. It actually might relate to dopamine signaling too. We're not sure at this point. What we also did here was we decided to look at dopamine-induced changes in function. We know from our previous studies that this stimulation of this pathway can evoke dopamine release. So we just asked, if we stimulate this pathway, can we see currents that will be altered via dopamine receptor antagonists? And so we blocked GABA transmission and glutamate transmission and just stimulated again at 20 hertz. And what we saw was that there was a number of different responses. Of course, there were cells that were non-responsive, but we also saw cells that had a depolarization and cells that had a hyperpolarization. And so the hyperpolarization was blocked with sulpirides, suggesting this was D2 dependent, whereas this depolarization was blocked with this serine 23390 compound, suggesting, providing a layer of support that this is a D1-like response. And so we have these dopamine responses, and so we just measured the amplitude of these responses in males and females, and we saw that there was a significant greater number of depolarization, or the size of the depolarization was greater in males compared to females, whereas the hyperpolarization was not different between the groups. However, when we looked at the representation across populations here, we saw that there was no difference between these. So there's some subtle difference here, but it does suggest perhaps some idea or some mechanisms how this might be altering function or differentially altering function in males and females. And so just to summarize this section, we found that chemogenetic activation was antinociceptive in male mice for both thermal and mechanical stimuli, but only mechanical in females, and only when we did this repeated sampling approach. This antinociception was absent following deletion of TA, suggesting it's dopamine dependent. I would say that this also agrees with the data that was published in eNeuro, where they used a pharmacological approach to show that this was dopamine dependent. So it was actually great to see a converging data set developing on this topic. When we looked at photostimulation, this was antinociceptive in both assays again. And really, when we started to probe and delve into the mechanism of how this can impact the circuitry, it immediately became somewhat complex. There was excitation, inhibition with sex differences in this polysynaptic inhibition and dopamine responses. So there's I think a lot more work to do there to try and decipher. And I'd say this is a very active area in the lab, and we're studying this. We're looking at plasticity circuits, as well as, how can we manipulate, or what compounds can alter signaling within the PAG as well as downstream in the BNST for some of these processes. So I wish we had more ready to roll on this, but we do have some emerging stories that I think will be pretty interesting and exciting. And so I will change pace now. So that was part one. I'll change pace now and look into part two. And I will say, when we started planning these studies, we envisioned one master one enormous paper that would kind of link these two things together. And I think what we realized as we got further or deeper and deeper into this subject is that this is a pretty complex area. And so we had previously, or I had previously shown that CRF signaling and dopamine have some sort of interaction in the BNST. We know that CRF neurons can regulate a number of behaviors in BNST, so we had found that they play a role in driving binge-like alcohol consumption, but also that local CRF neurons can drive a potentiated fear and anxiety due to acute SSRI exposure. And this was some sort of engagement of a local microcircuit. And from a pain perspective, data from Ide et al. In 2013, this is from the Manani group, showed that CRF can induce a condition place aversion in the BNST. And more recently, they had a paper demonstrating that in a model of chronic pain, that they had data suggesting these CRF neurons were hyperactive, so an increased firing rate, and this was reflected with an increased inhibition of these outputs to the VTA that others have shown as anxiolytic and stress buffering. And this was exciting because we had previously shown that with this aversive state with serotonin, we have a similar response, and as well as chronic alcohol exposure engenders a similar circuit response. So it's kind of interesting to think that this local microcircuit that's engaged in the BNST plays a role in driving changes in affective behavior. And so the way we approached this was two-pronged. One was asking, does local CRF in the BNST contribute to these pain-related behaviors, and then looking at the in vivo dynamics of these BNST CRF neurons. And so to ask the first question, we used a genetic approach. We have a floxed CRF mouse, courtesy of, again, Larry Zweifel, and we used an AAV approach to delete this locally in the dorsolateral BNST. And this slide just tells you that this is effective at reducing CRF expression in BNST in both males and females. And we, again, looked at the same behavior, Hargreaves and von Frey, looking at thermal nociception and mechanical nociception. And we could see that deletion of CRF from the BNST led to an anti-nociceptive effect in the Hargreaves. So this is a measure of thermal sensitivity, as well as the von Frey, a measure of mechanical threshold. So we see that when we delete this, we can get alterations in pain-related behavior. So it was consistent with some of the data from the Manani lab, at least suggesting that CRF and BNST plays some sort of role. However, I will note they focus primarily on affective measures of pain. And that's something we haven't really done enough yet to talk about. And so it told us that we're on the right path to look at the dynamics of these neurons during pain-related behaviors. And so to do this, we use an in vivo calcium imaging approach, CRF-Cre mice, putting a predependent GCAMP6S into the BNST, using an Inscopix approach, targeting the BNST, using a grid lens to target the BNST, allowing appropriate recovery and recording during behavior. And I'll say that Wayland used the Inscopix data processing approach, which is a PCA-ICA approach to get the responses that we're going to look at. And early on, we did some comparisons, and this seemed to be worked well for what we were doing. And so the question here was pretty straightforward in a sense. We just wanted to know, are these BNST CRF neurons activated during noxious heat exposure? Essentially, we conducted the Hargreaves assay. And so the lens was implanted in the right BNST, and then we did all of the trials, 4 trials, and then we did left, right, left, right. So 1, 2, 3, 4, alternating the side with a 10-minute inter-trial interval, and this is just a heat beam applied to the hind paw. You can see that's what it might look like. And so the question is, are these neurons activated during this noxious heat exposure? And so this is 49 cells at 4 trials. And so this is 7 to 30 cells per mouse. This is 3 male mice. And you can see that this white line indicates the heat, when the heat turns on. You can see that there are some cells that are responding here. And then when we looked in females, this is 68 cells, 4 trials per animal, 8 to 19 cells, and we had 5 female mice for the experiment. You can see again, there's some indication of heat responsive cells, and there's something more to it. And so one of the things we did with this is that was aligned to heat on, and we also, just in trying to understand this data, aligned to the heat ending. And you can see here that, again, there are some responses that are activating that appear to be stopping when the heat turns off. And so, the idea is that there's some sort of temporal property here, but we had to analyze this in a rigorous way. And so the way we approached this was defining kind of 4 phases of our heat exposure. We had our baseline, where we just took baseline measurements this is for z-scoring the data. We have our before period, which was 1 minute. We had our after period, which was 1 minute. And then we have the heat that is split in half with the start and end. Self-explanatory what the start and end are. And the reason we had to do it like this was we actually, we allowed the mice to behave and withdraw their paws. And so there is a variable time of the heat exposure. We thought this was important because we wanted to see the relationship of the heat exposure to the paw withdrawal. And you can see here, this just shows the average time of paw withdrawal. So the white bar is heat on and the green bar is the heat off. And we see an average of all 4 trials per animal here. And so what Wayland did was just rigorously analyze this, and this is average data just for visualization. When he analyzed this data, looking at the average z-score in these different epochs, he actually averaged each individual trace for each individual trial. And so we get a series of responses that looks like this. And I would say what we saw that in the male mice, we see that there was a significant increase in the average z-score for the heat start epoch, whereas in the female mice, there was a significant increase in the heat end epoch. So it suggested on face value that there was a difference between how male and female, how BNST-CRF neurons respond to this noxious heat in male and female mice. We wanted to look a little bit more at this and look at this across trials. And so this, again, this is the responses in male and of BNST-CRF neurons in male mice split across trials with the white bar, with the white bar being the heat exposure. And what we looked at was just the type of responses we saw, the amplitude of these responses, and then the latency of these responses. And so what we found was that when we looked at all of the responses for male mice, we get about 72% that exhibit a positive modulation, about 19% that have a negative modulation, some that don't respond. And this was consistent across all 4 trials. So we saw a very consistent, stable responding across trials for the male mice. When we looked at the amplitude of these responses, we saw this interesting data where there was a significant reduction of the amplitude of response over time, which suggests that maybe there's some sort of attenuation of this signal, perhaps some sort of desensitization of the response. And when we looked at the latency, we saw this really interesting difference between trials 1 and 3 and 2 and 4. And what this mapped onto really nicely was that we were looking at left versus right. And so we have an ipsilateral contralateral difference in the latency to this response. So this was interesting and perhaps not all that surprising. In female mice, we did the same sort of analysis. And what we found was that this was the overall response for all trials in females. However, it didn't look as stable between trials. And so this is consistent with the idea that there's a different, there's a fundamentally different response between male and female BNST CRF neurons from male and female mice. Again, when we look at the amplitude, somewhat consistent across trials, and the latency, again, there were some differences across trials. And interestingly, this doesn't really map onto the same sort of latency we saw in the CRF neurons from the male mice. And so to summarize this, we found that BNST CRF neurons in male mice are more robustly and consistently activated during thermal stimuli, and that the amplitude of these responses to heat reduces as trials progress. And whereas in the female mice, what I can say with some confidence is that it looks different right now, and we're still trying to understand why this might be different. And there's a wealth of data that suggests that there are sex differences in the BNST, as well as there are sex differences in how CRF itself can signal. So it might not be surprising that we're seeing these, or it's not surprising that we're seeing these differences, but we really want to try to understand the mechanism. And so when we think about how this might be integrated for some sort of model of how this works, I find that the study from Manami group is really informative. So they suggest that these local CRF neurons in the BNST exhibit a hyperexcitability during chronic pain. And so what we're seeing is during this acute noxious thermal stimuli, we see engagement of these CRF neurons, but we don't see a persistent engagement. There's this kind of time-locked activation that goes back down during baseline. And if anything, over repeated trials, we see a reduction. So this, to me, feels like a normal physiological response to a noxious stimuli, at least in focusing on the male mice. However, there might be an interesting shift in how these neurons work as we transition from acute to chronic pain states. And this is something we're really interested in studying. And so the next question we have is, how do these PAG dopamine neuron inputs come into play, and how do they abrogate function? And so obviously this is hand waving. We don't have data for this, but we're trying to speculate how this might work. Is perhaps we saw some complex interactions between neurons with dopamine neuron stimulation, BNST, this photostimulation. Perhaps what we're seeing is that there's some sort of the CRF neurons perhaps have D2 located on them, and that provides a brake on their activity. So activation of these dopamine inputs can suppress activity of this so-called noxious heat responsive CRF neuron, whereas we have these other neurons in BNST that project out to the VTA. Perhaps we have a D1 signal on these neurons or a direct glutamate signal on these neurons that's gating this behavior and somewhat acting as a functional blockade of this engagement of local CRF signaling. And perhaps there's some sort of local inhibitory circuit that's engaged there. And so this is something, it's a really provocative model that we have at this point no data for, but we're really excited to follow up here. And with that, I will say thank you for your time. And again, this was some great work from Waylon Yu, a graduate student lab. I would say thanks to the entire CASH lab for being a wonderful group of hardworking, smart people to work for, Larry Zweifel, for our collaboration to this, as well as the funders that have contributed to the funding to support this work. Thank you very much.